Hello everyone, welcome to my channel Bunny's Biology. Today we will learn about the mismatch repair system, that is MMR. Mismatch repair system is a post-replicative DNA repair pathway that corrects error during DNA replication, such as mispair bases or small insertion or deletion loop, thereby maintaining genome stability. So this is the correctly pair bases forming a stable double helix. In the first round of replication, a mismatch occurs. An incorrect base pair is formed, that is A pair with G instead of T. This mismatch is a replication error that can be repaired by mismatch repair enzyme or left unrepaired, leading to mutation. In second round of replication, where mismatch is not repaired, then the error become permanent, where one daughter strand retain the correct basis, that is A pair with T, but other inherits the mutated basis pair, that is C pair with G. This is now a fixed mutation in the genome. So to stop replication mistake from turning into mutation, cell rely on mismatch repair to fix the error in time. So what is mismatch repair mechanism? In E. coli, mismatch basis is recognized by MutH. MutH protein, that is homodimer form, scan newly replicated DNA. This is the newly replicated DNA or newly synthesized DNA and bind to mismatches basis pair, that is G instead of A. MutH embraces the mismatch containing DNA, inducing a nick in the DNA. The complex of MutH and the mismatches containing DNA recruit MutL and MutH and forming MutS, MutL and MutH complex. Next, MutH has endonuclease activity. So MutH has endonuclease activity that causes an incision on nick on one strand near the site of the mismatch. This is the mismatches form and a MutH is nicked in the site of mismatches, but the nick is introduced in the unmethylated daughter strand. So normally in E. coli have an enzyme, that is Dam Methylase, also called DNA adenine methyl transferase that methylates the adenine residue on both strands of the sequence 5' GATC 3'. This is the parent strand and this is the 5' to 3' direction and this is the 3' to 5' direction. So where 5' GATC 3', this is the 5' GATC 3', where 5' GATC 3' adenine residue is methylated by Dam Methylase. And the another strand also, this is 5' GATC 3' side and adenine is methylated by Dam Methylase. So the GATC sequence is widely distributed along the entire genome and all of these sites are methylated by Dam Methylase. So during replication, when a replication passes through the DNA that is methylated at GATC site on both strands, the resulting daughter duplexes will be hemimethylated. This is the daughter duplex strand and this strand is hemimethylated. Hemimethylated means one strand is, that is parent strand is methylated and daughter strand is not methylated. That is called hemimethylated. So this hemimethylated daughter strand is cut by MutH. So thus for a few minutes until the Dam Methylase catches up and methylates the newly synthesized strand, this MutH, MutL and MutH complex translocate on the DNA duplex until it reaches a hemimethylated GATC sequence. So the unmethylated or hemimethylated newly synthesized GATC sequence is cut by MutH because MutH has endonuclease activity. So then UVRD helicase unwind the DNA starting from the incision. So this is the DNA strand. So DNA is helix around each other. So this is the incorrect basis and this is the cut side. So this strand is cut by MutH. After cut by the MutH, this helix is unwound by the UVRD and unwound starting from the incision. And moving in the direction of the site of mismatches. Then exonuclease progressively digests the displaced single strand. But different exonucleases are used to remove single-stranded DNA between the nick and the mismatches. And depending on whether MutH cut the DNA on the 5' or the 3' side of the misincorporated nucleotide. If the DNA is clipped on the 5' side, this is the 5' side of the mismatch, Then exonuclease 7 or REC-J, which degrades DNA in the 5' to 3' direction. This is the 5' to 3' direction. Then remove the stretched DNA from the MutH induced cut through the misincorporated nucleotide. But if the cut or nick is on the 3' side of mismatch, this is the 3' side of the mismatch, then removed by exonuclease 1, which degrades DNA in 3' to 5' direction. This is the 3' to 5' direction. After removal of mismatches bases, this gap is filled by DNA polymerase 3 and sealed by ligase. So if I summarize, then first MutA scan the mismatch bases, then MutL, that coordinate repair system, and then MutH clip the unmethylated daughter strand, then UVR de-unwind the DNA, then exonuclease remove error containing DNA, then DNA pol 3 synthesize DNA, and then ligase seal the nick. This is the prokaryotes mismatch repair system in E. coli. This is the prokaryotic mismatch repair system. But in eukaryotes, mismatch repair mechanism is more complex than prokaryotes. In mammalian cell, mismatch is recognized by one of two MutS homologous. So MutS homologous. MutS homologous is two types. One is MutSalpha, another is MutSbeta. So MutSalpha, that is heterodimer of MSH2 MSH6, and MutSbeta heterodimer of MSH2 MSH3. So MutSalpha, that detects single bases. So it detects single bases, that is single mismatches bases or small loop that is up to 3 nucleotide in size. But MutSbeta, that detects larger indel up to 13 nucleotide size misincorporated bases. So 13 nucleotide bases. So MutSbeta or MutSalpha first scanning the mismatches bases. So after MutS and MutSbeta recruit MutLalpha. So then MutLalpha, that is heterodimer of MLH1 and PMS2 recruit in the side of MutSalpha and form a tetrameric complex. Tetrameric complex. Because MutS or MutSbeta have two heterodimer and MutLalpha have heterodimer form. So total is tetrameric complex. So MutLalpha has a endonuclease activity that car the daughter strand near the mismatch site. But daughter strand is recognized by PCNA, a sliding clamp that help identify the newly synthesized strand, and RFC that help in loading the PCNA in DNA. So DNA bound PCNA help MutLalpha identify and card the daughter strand at the mismatch site. After the incise the DNA by MutLalpha, exonuclease 1 is recruited and activated by MSH2 or MLH1. So activated exonuclease 1 remove the error containing DNA stretch. Then this gap is filled by DNA pol delta and nick is sealed by DNA ligase. So if I summarize in eukaryotic mismatch repair system, first MutSalpha or MutSbeta recognize the mismatches bases, then MutLalpha that have endonuclease activity that nick the daughter strand at the site of mismatch bases, and PCNA that help identify the newly synthesized DNA, then RFC that help the PCNA loading in DNA, then exonuclease 1 that remove mismatches bases, and DNA pol delta that synthesize new DNA strand, and then DNA ligase seal the repair strand. This is the mechanism of eukaryotes. This is the mismatch repair system in eukaryotes. This is the prokaryotes and this is the eukaryote in mammalian cell. So if the mismatch repair mechanism is fail or mismatch gene have loss of function of mismatch repair gene, then form microsatellite instability. So normally mismatch repair protein corrects replication error in this region, but when mismatch repair fail or mismatch gene have loss of function, then error accumulate and microsatellite become unstable. That means This insertion and deletion repeat expand, and this is called microsatellite instability, and that causes high mutation rate, and that causes tumor cell. And in the defect of mismatch repair, like genetic mutation or mismatch repair gene have epigenetic silencing that causes Lynch syndrome. This is the colorectal cancer, and this colorectal cancer often without many polyps, and they are developed in young ages, and that causes by the inherent mutation in MMR like MLH1, MSH2, MSH6, PMS2. So the mismatch repair system is a fundamental DNA repair pathway that safeguards genome stability by correcting replication error. Its proper functioning prevent mutation.